Major finding in this first phase did involve a gene that we knew was altered in DNA methylation in cancer, and actually had been discovered by our group to be so in 1998. And in the year 2000, we discovered that abnormal DNA methylation in this gene, which turns it off abnormally, actually sensitizes brain tumors to the most common and efficacious therapy that we have today, so-called alkylation therapy agents, the drug known as temozolomide. And so when you have the abnormal DNA methylation of this gene, which we call MGMT, it actually is a favorable prognostic sign for patients because their tumors are more sensitive to damage by the alkylating agent. So we knew that to start with. But what came under new perspective here was in the integrative power of this cancer genome project when we took our DNA methylation findings, which is the epigenetics, and matched it with the genetics being studied by other investigative groups in the project. And what we learned to our surprise is that when the tumors recur in these patients after therapy, the therapy does extend the life of these patients. But unfortunately at this time, in most all cases, the tumor will come back. That those tumors that had this abnormal methylation of MGMT and had lost its function now had a very increased number of genetic alterations or mutations in many, many genes, far higher than patients who had never been treated at all. And we think that this may lead in the recurrent tumors to their being more aggressive and being resistant to therapy. So there's a good and a bad to this. Loss of this MGMT sensitizes patients initially so they do better, but it also sets up a scenario when they recur that it may help drive the tumor. It's still a good therapy up front. It's still what we should use up front. But now this gives us a chance to potentially think, how can we deal with the bad side? How can we determine how we may, either from the initial therapy or when the tumor recurs, come to treatments that can deal with this bad side of the coin for use of such therapies. This also may be relevant to many other cancers where we use chemotherapy, where we induce these kinds of changes, and we may be able to be smarter about how to deal not only with brain tumors, but other types of tumors with the therapies we use today because of findings like this.